Okay, so in this video, let us now look at the epidermis. So recall that we have two layers, two main layers of the skin, which is the epidermis, which is the more superficial, and then we have the dermis, which is the deep layer. Right. So in this picture, you can see the epidermis begins from this layer upwards, and then the dermis is the lightly staining layer, and which goes downwards. Okay, so the epidermis is made up mainly of 4 layers, but however there's an exception here. We have a 5th layer which is present only in thick skin. Okay, so the 4 layers or the 5 layers of the epidermis from the basal layer going superficial are, we have the stratum basale, this layer here, the most deep layer of the epidermis is the stratum basale. We also have the stratum spinosum, which is the next layer. Then we have the stratum granulosum, which is this 3rd layer, this dark staining 3rd layer here. Then we have the stratum lucidum, this light staining eosinophilic layer here. So this is the exception. We only find stratum lucidum in thick skin. We do not find it in thin skin. We will define in the next slides what we mean by thick skin and thin skin. Okay, so then the most superficial layer is the stratum corneum, which in this preparation is the thickest layer of them all. All right, so remember we said we have dermal epidermal interdigitations which attach the epidermis to the dermis, and then these are made up of the epidermal pegs, this one, and the dermal papilla, this one. So they function so as to keep the epidermis attached to the dermis. All right, so the cells which you'll find in the epidermis include the keratinocytes, melanocytes, the Langerhans cells, and the Merkel cells. All right, so now let's look at the stratum basale. This is the deepest layer of the epidermis, and it is a 1-cell thick layer made up of basophilic cells. So these cells, they appear basophilic due to the fact that we have numerous ribosomes within the cytoplasm, and then these ribosomes are the ones which synthesize the keratins. Alright, so these cells, they are attached to the basement membrane. Recall that we have a basement membrane at the junction of the epidermis and the dermis. So the cells of the stratum basale are attached to the basement membrane by hemidesmosomes, and then they are attached to each other by desmosomes. Okay, so this layer is the one which has an intense mitotic activity, is the one which generates all the other layers. Okay, and so that's for this reason it's also called the stratum germinativum by some texts. In this layer we mainly find keratinocytes, melanocytes, and the Merkel cells. Right. For the stratum spinosum, it is a several cell thick layer, the next layer to the stratum basale. Okay, it is mainly made up of keratinocytes, and we may find some Langerhans cells in this layer. So it is made up of large polyhedral cells which have central nuclei, but these cells, they stain lightly eosinophilic due to the fact that these cells, they are synthesizing keratins, and then these keratins, they are assembled into what you call tonofibrils, which will fill the cytoplasm of these cells, making them appear slightly eosinophilic. So the deepest layers in this stratum spinosum, they show some mitotic activity, that some texts say that these deepest layers plus the stratum basale, they form the stratum germinativum. Right. So an interesting feature here is that these cells, they present with spinous processes between individual cells. So if you can look closely, you find that in the intercellular spaces we have little spines projecting These are the desmosomes holding the individual cells together. So they are apparent here because during the preparation the cells shrink a little, making these desmosomes visible. Now for the stratum granulosum, this is mainly a 3 to 5 cell layer thick, and it appears deeply basophilic due to the abundant granules present in its cytoplasm. That's why it is termed stratum granulosum. It is granules in its cytoplasm. So these granules, they're called keratohyalin granules, containing mainly trichohyalin and filaggrin. This trichohyalin and filaggrin, they will facilitate the aggregation of keratin filaments, facilitating the process which we call keratinization of the cells. Okay, so this is the point of terminal differentiation of keratinocytes, where they will begin their process of apoptosis and conification. Right. By that we mean that these cells, they will lose their nuclei, they will lose their granules, and they will lose all their organelles, and they become filled with keratin filaments in their cytoplasm. Right, meaning that they are now dead cells, dead keratinized cells. Okay, so another interesting feature of the stratum granulosum is that it contains the epidermal water barrier. Right, we'll talk about this when we talk about the cells making the stratum granulosum. So now for the stratum lucidum. The stratum lucidum is only present in thick skin, and it is a clear or a translucent layer, as you can see here. This layer, this one is the stratum lucidum, the clear layer between the stratum granulosum and the stratum corneum. Okay, so these cells, they are dead, flattened, anucleate, and keratinized. Okay, the stratum corneum is the outermost layer, the most superficial, and it can be 15 to 30 cell layers thick. Right, it contains squamated and fully keratinized cells, which will begin to undergo desquamation at the topmost surface here. The cells will be sloughed from the surface. Right. In this picture, you can see that the stratum corneum appears to be the thickest layer of all layers of the epidermis. But however, in most of the skin, the stratum spinosum appears to be the thickest layer, especially at the regions of the epidermal pegs or the epidermal invaginations here. Alright, let us now differentiate between thick skin and thin skin. Right, so this differentiation is mainly based upon the thickness of the outermost layer or the thickness of the epidermis. All right, so thick skin, it is a thicker epidermis as compared to thin skin, which is a thinner epidermis. If you can see in this picture, this is the epidermis of thick skin, and then this is the epidermis of thin skin. So thick skin is much thicker epidermis, and there's a thicker stratum corneum, and the stratum lucidum is present in thick skin, whereas in thin skin we have a thinner epidermis, we have a thinner stratum corneum. A stratum lucidum is absent, it's not present. So now for the dermal epidermal interdigitations, they're much more pronounced in thick skin because it is this skin which faces the most pressure. For example, the skin of the palms, it has to withstand a lot of pressure, so these interdigitations, they need to be as strong as possible. Thus in these areas they present with what we call fingerprints or dermatoglyphs. Alright, whereas in thin skin they are present but they're not that well pronounced. So thick skin covers the palms and the soles of the feet, okay, and then thin skin is found almost everywhere else except the palms and the soles. right? But however, for thin skin, is much more thinner on flexor surfaces than on extensor surfaces. So consider, for example, the elbow joint. The flexor surface is that inner aspect of the elbow joint where we have the biceps muscle and the cubital fossa. And then the extensor surface is the opposing surface where we have the olecranon process. Now, thick skin is hairless, whereas thin skin contains hairs except for the skin of the lips, the clitoris, and the glans penis. So basically, this is all about the epidermis. We'll see you in the next video on the dermis.